Welcome back. Today we are going to be looking at kind of the next progression of inheritance patterns, and we're going to be looking at non-Mendelian inheritance patterns, meaning these are going to be patterns that differ from the ratios that Mendel would have suggested from his observations. So they're going to be a little bit different than some of the problems that we looked at last week. So again, we're going to be looking at some examples. I'm going to do some examples here for you. Let's see if we can get the camera to focus in like it should. There it goes. So we're going to be looking at something we call non-Mendelian genetics. Right, and they're non-Mendelian genetics because they're going to result in ratios that differ from what we would have expected to see with Mendelian genetic patterns. So these are typically more complex, so we'll call them complex patterns of inheritance. So complex patterns of inheritance, and we're going to look at 8 varieties of complex patterns of inheritance. And we'll just kind of go through them. Today we'll look at maybe 3 of them, and then tomorrow we'll look at the remainder. So the first one we're going to look at here is called incomplete dominance. Incomplete dominance. And a very simple example for incomplete dominance is something that we'll look at here. So let me go ahead and share my other screen. And we're going to be looking at, look at this example here. So what you're looking at here are what we call snapdragon flowers. And we have a red snapdragon flower and we have a white snapdragon flower. The red allele is not completely dominant to the white allele. So instead of getting red or white flowers when they breed, like we would have seen with the peas, instead we get this in-between phenotype, this pink phenotype. And you can see that the symbols that we use are a little bit different. Instead of using capital letters and lowercase letters, we're using superscripted letters to show that it's an incomplete, or that it's incomplete dominance, that it's a non-Mendelian inheritance pattern, that it's more complex. So we use these superscripts when we're trying to show that it's more complex than just dominant or recessive. So again, these are snapdragons, and in this case, the red is incompletely dominant to the white, producing the pink. So we'll go back to our notes setup here, and we'll define this, and then we'll go ahead and work a problem in this arena. So we'll say that one allele doesn't completely mask the action of the other. So what we end up with is this alternative, this third phenotype. So we get this third phenotype. And a good example, again, are these flowers, these snapdragon flowers. So we get these snapdragon flowers. Good example of this. So let's work an example. Slide the paper up here. So I'm going to have, let's go ahead and just define our alleles. So we're going to use F for flower color. And then I'm going to use the R, the superscripted R, as red flowers. So red flowers. And I'm going to use an F with the superscripted W as my symbol for white flowers. So, and then obviously we're going to combine this, we're going to get pink flowers. And again, you got to remember that each plant is going to have 2 alleles. So one allele from each parent. So right now you're just looking at one allele and one allele. In reality, a plant's going to have 2 alleles. So let's do a cross. We're going to take red flowers and we're going to go ahead and cross them with pink flowers. Okay, so if we look at our genotypes, the red flower is going to be F, R, F, R, and the pink is going to be F, R, F, W. So the two alleles for the red flower, and then the pink is one red allele, one white allele. All right, so let's set up our F1, and this becomes very, very similar to kind of how we've worked this in the past, where our F1, we're going to set up our Punnett square. That's not any different. We're going to go ahead and put one parent on this side, and the superscripted letters need to stay with the alleles. We'll put the other parent up top, just like that. All right, and then we just fill in the box. Right, so all I've done is I've dropped down the alleles, and now I have my odds. So my ratio or my results, my odds set would be this one is going to be red and this one's going to be red because they have both red alleles. So I have two red, and then both of these are pink. So I have two pink. So two red, two pink. So other things that will show this type of inheritance patterns will be like chickens. So chicken feather color will be inherited with some incomplete dominance. The length of a dog's tail actually will kind of adhere to this pattern of inheritance. Eye color has some incomplete dominance to it. We'll see that again later. And then also hair texture, straight, wavy, curly, got some incomplete dominance at work there. Okay, so that is incomplete dominance. You'll see some of these in a problem set that will be posted this week. Next, we're going to look at something called codominance. So we'll go ahead and make a note here. Codominance. And we'll go ahead and share our screen, and we'll look at the example. So we'll go back. Let me go ahead and share my screen. And I want this. So here we have white cattle and we have red cattle. And when we breed them, we don't have a pink cow, but we have this roan cow that shows both the white and the red hair being equally expressed. So they're not incompletely expressed, they're equally expressed. And we see the white and the red hair on the cow equally expressed. So this is what we call codominance. So let's go ahead and show how we would work this. So we'll go back to the notes here. And we'll just make, again, a quick mark where we'll say codominance is different because both alleles, so both alleles are equally expressed. So we'll go ahead and work this example. Let's say that the CR is the red and the CW is the white. And let's go ahead and take these cows. So let's take a roan cow. So CRCW, and let's cross it with, again, this is a roan cow. Let's cross it with another roan cow, CRCW. And let's look at the results that are going to be projected based on this F1. So set up my Punnett square in the same manner that we've been setting them up. And I'm going to have one parent over here, so CRCW, the other parent here. And now I just fill in. So my ratio, my phenotypic ratio, I'm going to have one red, two roan, and then one white. So I have a 1 to 2 to 1 ratio, or 1/4 are red, 2 out of 4, 50% are roan, and then 1 out of 4, 25% are white. Okay. Other things that are inherited in this fashion, we're going to see, we'll do quite a few problems with blood types. So with blood typing, we have these antigen A and antigen B, and those are going to be codominant where the alleles are equally dominant. So let's get a look at that. Share my screen again. So here we go. Now we're looking at this ABO blood group system, and we can see these A antigens on the red blood cells that are type A variety. And what those are, they're just carbohydrate extensions from the red blood cell. Here we have the B antigen on the type B. So we have an allele for type A and type B, and they are co-dominant with one another. If they're both present, if you have a chromosome with type A and a chromosome with type B in the same individual, then you have type AB blood, and they are equally expressed. So the red blood cells have both variety A and B. Now, the ABO blood grouping system also has a different sort of inheritance pattern called multiple alleles. We have a mutant allele, a third allele, that codes for no antigen, and we call that type O, which is like a null set antigen. So we'll see that when we talk next time. So that's interesting. So back to the notes here. Then finally, we have what's called sex-linked or X-linked, and we're going to go ahead and give this a look. So this is sex-linked or X-linked inheritance patterns. And one that's very familiar and common is going to be this red-green color blindness. So let's go ahead and share my screen again. And what you'll find is X-linked alleles are more prevalent in males than females, because males, they only have one X chromosome, so they're going to have it or not. Whereas females, they have a chance to mask a recessive allele on the X chromosome. So for instance, let's get a look at, so here's your typical red-green color blindness test, and they'll use this just to determine if someone has this red-green color blindness where they can't see the difference between red and green, or to really make that difference out. And that is a mutated allele that's carried on the X chromosome. So about 8% of the population in males will have red-green color blindness compared to 0.4%, 0.4% of females. So it's much more prevalent in males than females. Hemophilia, the inability of blood to clot, is also something that can be a mutant allele on the X chromosome that males have way more often than females. Also, male pattern baldness is something that's carried on the X chromosome. So again, this red-green color blindness test, you can see these the red background with the green digit inside, or vice versa. Somebody with red-green color blindness would really struggle to make out the digits against the background here. So let's talk about how we work with this. So let's go back to the notes. And what we will say is these traits are carried on the X chromosome. So we'll say these are traits carried on the X chromosome. And we know that females have the genotype XX, and males have the genotype XY. So when we show these alleles and we show these genotypes, we're going to have to show these X and these Ys, and we're going to have to show the gene or the version of the allele on the X chromosome. So again, we're looking at things like red-green color blindness. So red-green color blindness. And we're looking at things like hemophilia or male pattern baldness. So let's give this a look. Let's say that we've got a big R is normal vision, and a little r is this red-green color blindness, and we'll just abbreviate it there, red-green color blindness. So let's say that we've got a parent set, and we'll say that dad is colorblind, and he's going to mate with somebody, a woman, that has normal vision, so she's got a big R, but her father was red-green color blindness, or had red-green color blindness, which means the dad is going to have to donate this X to her, and she's going to then be a carrier. And they want to know what are the odds that they have offspring that might be red-green colorblind. So what we would do is draw our Punnett square and go ahead and bring down our individuals into the square, right? We just start filling it in. So females, it's 50-50. 1 out of 2 females are going to be red-green colorblind or normal. And males, it's going to be the same 50-50 as well. So 50-50 of the males will be red-green colorblindness. So if we were going to do a full phenotypic ratio, 1/4 would be normal males, 1/4 normal females, 1/4 red-green colorblind males, and 1/4 red-green colorblind females. So our ratio really would be 1 to 1 to 1 to 1. This be our ratio. That'd be our full ratio set. But if they asked us just about males or females, then we could talk specifically about males. So if they had a son, what are the odds that the son is red-green colorblind? Well, it's a 50% chance, right? And if they asked us just what are the odds that any offspring is colorblind, that's going to be 50% chance as well, because you take the half chance that, any sex is going to be 50%. The odds that it's a male and colorblind, you'd have to then multiply the 50% chance that it's male versus female, and then 50% chance that it's red-green colorblind versus normal vision, and then that's where we kind of get our quarter odds over here. So that's red-green colorblindness, and those are the first 3 in the complex patterns of inheritance. And we'll look at the remaining items in the next session. We want to try to keep these short. We're kind of running it. We're about 18 minutes here, so that's probably good for this one. So I hope that's helpful, and I'll see you next time.